And so now we'll be talking about how this organism utilizes macromolecules. It uses carbohydrates for growth and development as well as a source of energy, and its excess energy is stored in the form of starches. This gives the plant cell.
This diagram shows how carbohydrates are used as glucose units, which become microfibrils that become cellulose fibrils that make up the cell wall.
Now, the way it uses lipids, it makes the plant very resilient through a variety of conditions. It has like wax-coated leaves that basically show the plant's health, and also uses protection and provides its structural integrity, as well as it helps its soil health as it's planted in the ground. And the lipids are being pumped out from fungi and into the soil to provide nutrients for other plants as well. Now, the proteins it uses act as enzymes to catalyze different reactions, and it also provides added structural integrity and provides defense against pathogens. And they also act as storage mediums to meet growth and nutritional demands for their seedlings.
Diagram of proteins show how rubisco that acts as an enzyme to catalyze photosynthesis.
Nucleic acids allow the plant to grow and reproduce by storing its genetic information, and promotes differentiation among its plant cells and tissues. And they store and transmit genetic information through the linear sequence of nucleotides in their DNA.
It shows the difference between DNA and RNA. Focuses on organelles, membrane structure, and transport mechanisms. The organelles of our plant are pretty much the same as a regular cell, plant cell. But there are some small adjustments. For example, there are more mitochondria, which support the more energy uses for the adaptations of the plant. There are more transport proteins in the membrane, more ion pumps, and more flexible, thinner cell walls. For the membrane structure, since the cells around the mouth are adapted to basically breathe as mouth, the increase of ion pumps in transport membrane or transport proteins allow the mouth to diffuse gases more easily. The flexible membrane also allows for the mouth to open and close. The pumps allow the mouth to open and close, and the more mitochondria give increased energy. For the transport mechanisms, there are three. There's diffusion, where CO2 enters and O2 leaves by protein channels as the gases move from high concentration to low concentration with no ATP required. And this happens as the mouth is open. Number 2 would be active transport, which controls the mouth movement. As K plus is pumped into and out of the mouth cells, it causes osmosis. And for 3, osmosis, depending on the ion concentration, water moves through the partially permeable membrane through aquaporins, and changes the cell shape. If the cells are turgid and they're full, they will open the mouth as they're firm, and then the cells, when they're flaccid, it will close the mouth. For Unit 3, cellular energetics and energy acquisition, sunlight goes into the leaf, and from there it goes to the thylakoids that are in the stoma. This diagram shows how the process of photosynthesis and cellular respiration, as it uses enzymes and ATP, as it goes through the Calvin cycle and cellular respiration with the enzyme rubisco that catalyzes it.
Now we're going to talk about how this organism navigates through cell communication and the cell cycle. Now, the signaling pathway for the evil convallaria is pretty particular to that of a plant, where the process goes as such. Signal reception, where the stimulus is detected by a specific cell receptor on the cell surface. The signal transduction, also known as the relaying, where the signal is converted and amplified through a cascade of events. Multiple pathways then interact, setting the stage for a coordinated cellular response. The signals will then reach the nucleus, which will activate specific genes, ultimately leading to changes in growth, metabolic activity, and even cell division. Now, evil convallaria contains both plant and animal cells, so the way it signals cells may be different from other plant-like organisms. However, it does act differently when around different species of convallaria. For example, nice convallaria has strictly animal cells, and if evil convallaria is nearby it, it will signal like how animal cells signal. For feedback loops, our organism is sort of a master of detection and can sense change in its environment and makes decisions based upon those changes. Its internal response would sound something like, should I grow more leaves? Should I open my mouth? Grow taller? And as a result would be any given action. If all goes well and the plant functions at a steady rate, it would be considered a negative feedback and would stop that action. A positive feedback, for example, that action would be further amplified for the plant to be able to get to a point of stability. The evil convallaria goes through a very similar cell cycle checkpoints that other plants also undergo, but it is slightly different. What stays the same for evil convallaria is the G1 checkpoint, which monitors cell size, availability of nutrients, and the DNA readiness before the cell undergoes DNA replication. Now, pausing and getting off track for a minute. A fascinating fact about evil convallaria is that they have abnormally large leaves for its size, and therefore the cell cycle is slightly abnormal to accommodate for that leaf size. This organism undergoes a process called indoor duplication, which occurs in the S phase of the cell cycle, where it replicates its DNA and skips division. This process is needed for evil convallaria due to it needing more structural support and a larger surface area to accommodate for the leaves. The result of this process is a cell with multiple copies of its genome with a single nucleus. This state is called polyploidy. The increased polyploidy is associated with a thicker and more robust cell wall, providing better mechanical support for a larger leaf structure, which helps it maintain shape and avoid tearing. It also creates a more resilient. Here's a rough breakdown of it. Normal cell cycle would go such as G1, S, G2, mitosis, and cytokinesis. The evil convallaria cell cycle, it'll go G1, S, skips G2 and mitosis, then back to the G1 state, but then the DNA is replicated.
Exchange in not eating food, because it cannot obtain energy from food. It relies entirely on photosynthesis and cellular respiration to live. When one of the energy pathways is disrupted in any way, the effects can be seen across multiple biological levels. At the molecular level, pathway distribution interferes with enzymes and energy transfer. Enzymes control each step of photosynthesis and cellular respiration. If these enzymes are inhibited or fail to function properly, reactions slow down or cease. As a result, glucose production or ATP synthesis decreases, reducing the amount of usable energy available to the plant. At the cellular level, reduced ATP availability impairs normal cell function. To maintain homeostasis, power active transport, and carry out life-sustaining metabolic processes, cells require a constant supply of ATP. Without sufficient ATP, the internal environment of the cell cannot be properly maintained. Long-term cellular activities related to growth, repair, and reproduction fail to proceed in an orderly fashion. At the level of organisms, such cellular failures have apparent consequences for the whole plant. As the mouth is used only for respiration, not for nutrient uptake, the plant relies entirely on efficient pathways of energy. Any disruption in the way energy is produced slows down the growth and limits the reproduction, making plants less capable of surviving in the environment. It may droop, weaken, or lose in competition for resources. This shows how a disruption in the energy pathways result in an upward cascade. The molecular changes related to enzyme activity translate into cellular energy deficiencies, which eventually affect the survival of the whole organism. This emphasizes how the continuity of life depends on the effective acquisition and utilization of energy.